We've already discussed 3 organic molecule categories. We've talked about carbohydrates, lipids, and proteins. We told you during the protein presentation that we were going to talk specifically about enzymes, which are a type of protein, in a separate presentation. Well, this is that separate presentation. So let's talk a little bit about proteins. Enzymes, sorry. Enzymes, all of their names end in the suffix ase. Usually the name tells you the molecule the enzyme acts upon. For example, amylase, which is the enzyme that acts on amylose, lactase, which is the enzyme that acts on lactose, and peptidase, which is the enzyme that acts upon peptides. I hope you guys all know what a peptide is because it was in the last presentation. If there are exceptions, meaning if their names do not end in ase, then that name that you are hearing is not their scientific name. An example of that, and actually what you're going to be graphing for homework over the weekend, are the 2 enzymes pepsin and trypsin. Those are not their scientific names. They are actually proteases, which tell you that they act upon proteins. Enzymes allow chemical reactions that normally occur in harsh environments to occur inside cells. As we know, our bodies are not harsh environments, so enzymes allow those types of reactions to occur inside of our bodies. Enzymes are affected by pH, temperature, and salinity. Everybody should know by now that pH is whether something is an acid or a base, and salinity has to do with the salt concentration. Enzymes, as I've already said, are a category of proteins. You guys should know the other protein examples and what they do. I'm not going to cover that now. Enzymes are biological catalysts, which means they are catalysts that are found in living organisms. As you can see, biological and found in living organisms are the same color. That's because that's what that means. What is a catalyst, you ask? A catalyst is any chemical that speeds up a chemical reaction by lowering the activation energy of the reaction. There are many different catalysts that are used in chemical laboratories. A catalyst, as we said, is something that speeds up a chemical reaction. If it is doing that inside a living organism, then that catalyst is called an enzyme. So all enzymes are catalysts, but not all catalysts are enzymes. Now you can see that we have activation energy in blue because I'm guessing you don't know what activation energy is. Well, let's find out. Activation energy is the energy needed for a chemical reaction to take place. I'm going to show you that in just a minute on an energy diagram. So, here's an energy diagram. This is where the this is where the reactants are. This is where the products are. This shows you the energy that's required for the reaction to take place. If you look at where the reactants are, and then you look at how high this curve is, or what we call the top of the hill, that amount of energy right there is the activation energy. This is an exothermic reaction. Everybody should know that's one of the words that's on your review of chemistry sheet that I gave you. Exothermic reactions produce heat, and endothermic reactions require heat. So if you notice on this one, this is the reactants and this is the products. The products have less energy than the reactants because during this exothermic reaction, heat was given off. In this energy curve, this is the reactants, this is the products, the activation energy, here's the top of the curve, so from here to here is the activation energy. Because the products have more heat than the reactants, this is an endothermic reaction and heat was absorbed. Here we have another energy diagram that has everything labeled. We have here the reactant energy. We have down here the product energy. From the reactant energy to the top of the curve, that is the activation energy. From the reactant energy to the product energy, that is the heat of reaction. This case, the reactant energy is higher than the product energy. That means heat was given off during the reaction. So would this be an endothermic or an exothermic reaction? Hopefully all of you said that it was exothermic. Every enzyme has a uniquely shaped active site. Once again, active site is in red because I am going to tell you what that is. The active site is the area on the enzyme where the substrate binds. The substrate is the molecule or molecules that is or are acted upon by an enzyme. I know that's kind of confusing, but once you understand what's going on, substrates can be single molecules or they can be multiple molecules. So we'll talk about that a little more when we do this in class tomorrow. The enzyme substrate complex is the name of the molecule formed when the enzyme and substrate bind together. So here we have a picture. This is the enzyme. This area here is the active site, and up here we have the substrate. So this enzyme is going to act upon this substrate. This here is the enzyme substrate complex. The substrate is already bound to the enzyme. That's where the reaction is going to occur. And then up here we have the products being released, and now the enzyme is free to repeat the reaction on another set of molecules. Once again, this is a different picture. You can take a look at that. Each enzyme can affect only one specific reaction or at most a few related reactions based on the shape of the active site. If you go back and look, you can see that this substrate fits exactly into that enzyme active site. On this one, you can see this substrate fits perfectly into this active site. This is known as the lock and key model of enzyme action. As you can imagine, if everybody's keys fit every lock, we wouldn't have any security in our world, would we? This is a diagram that just shows you a key going into it just like a substrate fits. This is the lock and key complex. This is the enzyme substrate complex. Just a kind of cool analogy. The components of an enzyme alter the substrate molecule just enough to weaken the existing bonds. The structure of an enzyme changes, but not permanently during a chemical reaction. As you saw on that first slide of the enzyme action, that enzyme was able to then go ahead and catalyze another reaction. I'll show you some more examples in a minute. After the reaction, the enzyme regains its shape and is free to affect other chemical reactions, but only if the active site is similar enough that the substrate will fit into the active site. So what this means is enzymes are used but not used up in chemical reactions. We're going to do a lab next week where you can see that enzymes can be used over and over and over again, and you will do that. Here we go once again. We have the enzyme. We have a uniquely shaped active site. We have a substrate who has a portion of the molecule that fits perfectly into that active site. This is the enzyme substrate complex. The bonds here are weakened slightly. This is broken into two different products. Now the enzyme goes back to its original shape so it can be used again. And here's a more realistic example because this is actually the substrate is sucrose. Here's the enzyme. This is the active site. We're going to do a worksheet that is very, very similar to this in class. It doesn't use sucrose, but it does use a molecule that's very similar. So tomorrow, when you're in class, we're going to do a worksheet. You will be able to label all of these different things going on. Take a good look at this because it will really help you when you're doing the worksheet. And that's it for enzymes. I'll see you tomorrow. Have a great evening.